So this is a component of a two-part project funded by OICR for developing diagnostic tracers for better breast cancer biopsies. One part of it that's done by our lab is development and validation of a novel positron emission mammography tracer. And the second component of this is something that's done by Aaron Fenster's lab, that was going to use this tracer for developing his biopsy guidance system. So the idea of this project was fairly simple. It was essentially to create a tracer that would be better than FDG for imaging breast cancer, to provide a high resolution image for that biopsy guided system, imaging guided biopsy system. It was very clear cut, the aims when we started. The idea was to use an antibody that had already been validated to target CD133. It had a chelator that we knew what it was going to be. It was going to be DFO. We had a positron emission tomography isotope, zirconium-89. We're going to put all of this together and synthesize it. The routine was as usual. We're going to do in vitro validation for specificity, affinity. We're going to look at internalization and KD values. Following that, we're going to do biodistribution studies and follow that up with in vivo imaging of a xenograft tumor model. We did all of that, and I'll show you some of that data. But by the time we got to the in vivo imaging of a mouse model, things started to fall apart a little bit, as things are normally. Nothing goes exactly as planned. But it wasn't all bad. It provided us with some interesting alternative approaches. And if there's time today, I want to sort of focus on that a little bit, sort of what our future approaches are going to be to address some of the issues that we encountered. Finally, the idea was to take this tracer and put it through clinical translation with our partners at McMaster at CPDC and have that ready for Aaron to use in his biopsy system. So a bit of a background on the target that we were going to use for imaging. The target is CD133. It's 120 kilodalton glycoprotein that's expressed on the cell surface. And it's a widely used marker for tumor initiating cells. It's associated with cells that have stem-like properties that have self-renewal, differentiation, and it's overexpressed in a number of cancers, not just breast cancer. There is a bit of controversy with using CD133 as a target, and part of that is because of the sort of established and existing antibodies that target that glycoprotein. In particular, AC133, which is widely used for looking at CD133, loses affinity in post-translational modification. So the good news was that through our collaborators, Jason Moffat at the University of Toronto and Sheila Singh at McMaster, we had access to a fully humanized antibody with very, very high affinity, sub-nanomolar. It was this antibody RWO3. And the other good news was that we had actually already used this RWO3 and radiolabeled it with a therapeutic isotope that we had used. It was lutetium-177, and we were using it for, the idea was to treat GBM tumors. And we had already seen some efficacy and specificity with this radiopharmaceutical based on the same antibody. So it was very reasonable for us to be optimistic on that simplified approach that I put forward on the aims that we had proposed on the OICR application. So we had a good starting place. So I'll share some of the data and sort of a journey that we're currently on, I suppose, still. The steps are very simple relatively. The idea is that we start with the antibody RWO3, add the chelator to the antibody, then attach the radioisotope, zirconium-89 in this case. We usually follow that up with validation to make sure that it's a new molecule. There's been some changes to the antibody. So we want to make sure that the affinity is retained and the specificity is retained. So we do all of that work in an in vitro assay. Once we have that validation and we know that our synthesis is good and we have a pure compound, we do some stability studies with that as well. And we follow that up with injection into a mouse model. And the idea is that with that mouse model, we want to make sure that it is expressing the target that we're going after. CD133 is not highly expressed. It is expressed um, in terms of triple negative breast cancers. There's literature to show that it's expressed in about 30%, which is good. But generally speaking, with any whenever you're going after a stem cell marker, it's not a highly expressed target. So you want to have that high affinity and specificity, which we had that with the RW03. We did some testing with a cell line that we had. It's a colorectal cell line. We did that both in vitro through flow cytometry, and also we grew that tumor in xenograft animals, and we showed that the expression of CD133 was still there. Mind you, if you have a sharp eye, you'll notice we used the AC133 antibody here for that purpose, not our RW03, just because we didn't have enough of it. But we did show expression of CD133, and we also showed that through modification of that antibody with the chelator, we still retained our affinity for CD133. And we did some IHC stains, and we showed that basically that the same thing, that our target was expressed both in vitro and from the excised tumors that we had in vivo. The next step would be just doing biodistribution studies. This is following our in vitro validation. I'm not showing all the data, but the first step would have been doing the synthesis, which we do, and validating that our molecular structure is in fact what we are assuming it to be through MS studies and LCMS. And NMR. So we have done all of that. The next step would be to inject it into a xenograft, and we did that with a tail vein injection. We let it circulate for a while, and then we take out the organs, and we, instead of imaging, this is equivalent to imaging, but we get an ex vivo count of where that radio-labeled antibody is ending up. And on the right here, you can see this is sort of our data from the therapeutic study with the same antibody with lutetium-177, which is a beta emitter. It's used for therapeutic studies. It also has gamma emission, so you could do some SPECT imaging with that as well. And we had some very encouraging results with that. We had very high tumor to blood uptake. After 96 hours, we furthermore achieved in vivo blocking, which was very nice, with a cold compound to show specificity. This is where we inject basically more of the cold compound to saturate what we're targeting. If you're saturating the target, there should be no more uptake of the radiolabeled tracer, and we did observe that. So specificity was confirmed. The data with zirconium now, with the same antibody, was okay and encouraging. This is again biodistribution studies in the same manner, inject tail vein, and we take the organs out at 24, 48, and 96 hours in this case. Still increasing tumor uptake, which is really good news, and decreasing blood. So that means that it's basically slowly going through circulation, getting excreted. And the tumor uptake was continuing to increase. So this is all good news so far and encouraging enough to go to our in vivo work. We're having some issues with our in vivo imaging at McMaster at the moment. So we did this work with our collaborators at Sunnybrook, and future work is going to be done with STAR. And unfortunately, this is not, if you're in the pet world, this is not, it's not bad, but it's not the kind of image you'd want to see. After you can still see that the uptake is increasing. If I have a laser pointer here, and you can see that if the tumor was in the flank, and you can see the tumor uptake is increasing a little bit, but there's still a huge amount of liver, and this is a very spotty image, not exactly what you'd want to see. Part of the reason for this, now we did not give up hope. This is so far the only imaging we've done as part of this OICR project. Part of the reason was that the logistics didn't go exactly as we had hoped. The tumors were not very big. We did not manage to inject enough of a dose. The tail vein injection didn't go so well. So we are repeating the study, and one of the actual biggest problems with it was that the number of chelators we had on the antibody was on average less than 1. And the mass that we had to inject to get enough of an activity to be able to obtain an image after 144 hours was significant enough to actually cause self-blocking. If you go back to the previous slide, You remember, we were actually intentionally trying to do self-blocking to show specificity. So if you have too much of a mass, you're going to get to this region of self-blocking. And that's part of what was happening here. So we're going to repeat the study as is exactly, but with bigger tumors, higher yield of our radiochemistry, better injection, and more chelators per antibody. However, it's not ideal. Even with this setting, we should have had a better image. So at the same time, when I say we're going to start looking at alternative approaches, this is not so bad, because it provides us with an opportunity to look at what's going on and what else we might be able to do. With the DFO chelation of the antibody, we're targeting the cysteine residues on the antibody. However, if you go with a DOTA chelator, and the DOTA chelator is what we had used with lutetium-177, we have the opportunity to go after a lot more residues. Mind you, of course, if you the more of those residues you conjugate with a chelator, you're going to lose affinity potentially. So that's something we're going to have to test. And another big issue with the DOTA chelation is that it's not necessarily a very good chelator for zirconium-89, and requires high temperatures for efficient labeling. Antibodies, while you could do that with a small molecule or even peptides, antibodies don't stand up too well with high temperature synthesis. So we need to find some alternative approaches. And just another sort of a structure of the macrocyclic chelator like DOTA on the right, and DFO on the left. One more advantage of the macrocyclic type of chelators like DOTA is that their stability. With the DFO zirconium-89, one of the other things we had seen both in biodistribution and the images was bone uptake, and that points to lack of stability of our radiopharmaceutical. If the zirconium or radioisotope falls off the chelator in vivo, it breaks down, it basically goes after bone, and it gets incorporated into bone, and that's not exactly what we're targeting. So we're going to have much higher stability with DOTA. The question is, how do we get it inside? And one of the expertise we have in our lab at McMaster is this idea of bioorthogonal chemistry and pre-targeted imaging. So what we're looking at here is basically putting half of bioorthogonal click chemistry, transcyclooctene, on the antibody, and we have the other half, the tetrazine, with our radiolabeled. Now this tetrazine could have the DOTA chelator on it, and you could heat this up, and this will stand to high temperature. So you could radiolabel the tetrazine, and then this way you could attach the two together without ever having to. And this reaction between the TCO and tetrazine is a very fast bioorthogonal reaction that happens at room temperature. So in this way, you never really have to heat up the antibody. That's one possible solution we're looking at. Another thing you could do, which is actually kind of a lot more exciting, is a little bit more challenging, but also exciting, is you could, in fact, put this antibody in circulation. Antibodies inherently take they're large molecules, and they take a very long time to circulate and find their target. And that's why we matched it initially with zirconium-89. The half-life of zirconium-89 would match better with an antibody as opposed to a shorter lived isotope like F18 that's traditionally used for small molecule imaging in PET. However, in this way, if we have this antibody with the TCO circulating in vivo, and we let it find its target and be excreted, and the patient is not exposed to any radioactivity, and a few days later, 3 days later, we could come back and inject the counterpart, the tetrazine, radiolabeled tetrazine, and that, because it's a smaller molecule, will circulate much faster, find its counterpart, the antibody, and we have an in vivo bioorthogonal reaction. That's a very fast reaction. We can do in vivo labeling and of CD133 in this case. It's a little bit more challenging, and there's, of course, there may be some questions that come up with this. We have shown it with different targeting moieties. We've shown it with bisphosphonates, and we're working to show this with the RWO3 antibody as well. We have synthesized, the other good news is on the chemistry side, we have synthesized the antibody, and we've managed to put 8 of these TCOs on the antibody. So this will help our specific activity. If you recall, specific activity was one of those issues that was a drawback with the poor quality of images that we had. Having 8 TCOs per antibody enables us to basically have an 8-fold increase in the specific activity, which in turn means we can inject 8 times mass to avoid the saturation of our target. The other big advantage here is that we have there's relatively new chemistry for putting aluminum fluoride inside of these NODA, again, microcyclic chelators. You could add aluminum fluoride inside. Aluminum fluoride is very, very exciting because F18 is by far the most widely available radioisotope. You can get it from any cyclotron. Basically, anywhere within North America, there's accessible FAT from a nearby cyclotron. We have cyclotrons at McMaster. There's cyclotrons all over Ontario now. And they have the problem is that obviously they have a 2-hour half-life. So with a 2-hour half-life, you can't directly put the F18 on an antibody, because by the time the antibody takes 3 days to find its target, all of the F18 would have decayed to background. However, using this bioorthogonal kind of approach and chemistry, we could have the antibody go find its target, and subsequently you just radiolabel the aluminum fluoride inside this NODA with a tetrazine, and you inject this, and this very quickly through a fast bioorthogonal reaction will go and find the TCO RWO3 antibody. We have synthesized this and shown its purity, and there's literature out there that actually shows the sort of the biodistribution and there's some pharmacokinetic data for a NODA aluminum fluoride tetrazine out there as well. So we can build on some literature that has already been reported. So that's exciting. And you know, a big advantage if you're looking at clinical translation down the line is it's always a challenge, not that it's not doable, but one of the things that's held back clinical translation of antibody work as a diagnostic positron emission tomography tracer is that you inject the patient with the isotope and they're going to have to go home, walk around with radioactivity inside of their system for 3 days, come back 3 days later and get imaged then. Whereas with a system like this, you inject the antibody without any radioactivity, and once you put the radioactivity, it's a shorter lived isotope that's been used for a long time in the clinic. So that's a much more, in terms of feasibility, clinically easier to translate. One more approach that we're looking at has nothing to do with radioactivity or positron emission tomography, but it's very similar in its targeting. It's a molecular targeting. Again, we use the same antibody and we use it with the same chelator, but instead of a DOTA radioisotope, we put a photoacoustic dye on there that we've developed. We have a novel one we've developed in our lab. Photoacoustics is where we basically use near infrared laser, which has a higher depth of penetration, so much more useful than fluorescence for in vivo imaging. And through thermal expansion and relaxation, the dye basically absorbs the near infrared optical wavelength and gives off an ultrasound. And we could use an ultrasound machine to pick up basically where this has been distributed. On this side, we've also synthesized and validated the synthesis and chemistry. And we have done some in vitro validation work as well to show that our RWO3 antibody conjugated with the dye still maintains affinity towards CD133. Other exciting opportunities, now going back to PET, but beyond the antibody, and remember the idea was to come up with a novel tracer, something better than FDG. One of the things that has been shown recently to be a driver of mammary carcinogenesis is inflammation. An induced form of an enzyme and inflammatory stimuli induces something called COX-2, cyclooxygenase 2, that takes arachidonic acid and produces a range of prostaglandins. And there's a lot of recent literature now that shows that these prostaglandins are a contributor to the early onset of breast cancer. So again, if we're going, just like stem cells, if we're going after early onset targets of breast cancer, looking at inflammation and particularly expression of this enzyme COX-2 would be very valuable. With work at UCLA, the challenge with this COX-2, we're not the first ones to go after it, but none of them really have worked in vivo, partly because it's really hard to radiolabel. If you could radiolabel it with an alkyl chain, that will work. You could put a carbon chain here and add a fluorine at the end of the carbon chain. That usually falls apart in vivo. If you put it on this pyrrole ring, it will be very, very stable, but that's a very, very difficult position to label nucleophilically with with a fluoride ion. What we've developed at UCLA is a electrochemical approach where you take sort of electron-rich moieties like this, you bring them onto the surface, you oxidize them, you create this radical intermediate that is much more receptive to something like fluoride ion, and you could radiolabel and have this fluorinated COX-2 inhibitor, which is very similar in its structure to celecoxib, a drug that was actually used and still is in use a little bit as an anti-inflammatory specific to COX-2. We have synthesized this, so that's another tracer that's available to us, and we've done some in vitro validation again in the same way. We've shown that it's specific to expression. Now this time we're going to show a specificity towards COX-2. So we take some macrophages and put some endotoxins in them, express cyclooxygenase 2, and then show affinity towards our tracer. And with increasing expression of COX-2, which we could do with Western, we see increased uptake of the tracer. And we could do blocking in the same way that I showed with lutetium. Again, with a cold compound, we show that we have lack of uptake when we saturate the same enzyme with a cold compound. So that's one option that we also have that we could try. Another one that's actually very exciting is and a hot topic. This is work we did with David Shackelford at UCLA, is targeting mitochondrial metabolism. We did this work looking at non-small cell lung cancer, and we found very, very interesting heterogeneity in the different types of tumors. Some that were very highly glycolytic, this is the heart, and some, this is FDG, so they were very reliant on glycolysis, Warburg effect with the traditional kind of tumors that we look at. And some that actually were reliant, not so much, they had negative FDG, but they have positive, this probe that we have, FBNTP, that looks at mitochondrial activity, oxidative phosphorylation. So there was two different types of tumors, and there is known literature out there that shows that triple negative breast cancer, which we're going after in this OICR project, has similar metabolic signature profile to non-small cell lung cancer. So that is to say, if remember our original goal was to find a tracer that's better than FDG, and if you have an FDG trace that you're looking at a tumor like this, that is not glycolytic, you would miss it completely. Whereas with this particular FBNTP tracer, which is basically a cationic tracer, it looks at mitochondrial membrane potential. You would have a positive, oxidative phosphorylation region, so you could pick up that that tumor is still active. And not only would that help you in your diagnosis, which Aaron can use for his biopsy guidance, you could also guide it to stratify patients who would benefit from a particular type of treatment. This patient, for example, would not benefit from an inhibitor of glycolysis, but would benefit from inhibitor of mitochondrial activity complex 1, for example. Leave it at that. If none of these things work, on the positive side, we have, through our partner CBDC, we have DCFPYL, which has gone through its clinical translation for PSMA targeting, and that is also available now. They're not using it for breast cancer, but that would be another tracer that could be available for breast cancer imaging. So we have a lot of options available to us, which is nice. That things didn't quite work out in the first try, which is, I suppose, the way research goes. But we are hopeful that this opens more opportunities than we had before. Thank you to our collaborators, Jason Moffat and Sheila Singh. They're providing us with the antibody and working with us on a therapeutic approach with GBMs. CPDC is our in-house partner. They provide Ontario with a range of clinical positron emission tomography tracers, and they're helping us with clinical translation of a number of tracers that we sort of touched on today. CIHR for funding this, for funding the therapeutic approach with GBMs, and OICR for funding this diagnostic approach targeting CD133. A very capable McMaster team that I've inherited recently, and of course John Valiant, who initially started this project with OICR. And I think I'm still on time, so I'll take any questions you guys might have.